Okay, let me introduce myself first. So I was born and grown up in the southern part of Japan and moved to Tokyo and graduated from Faculty of Medicine, University of Tokyo. And there I got my doctoral degree while working as a physician of hematology and oncology. And then I worked in Hiroshima for 3 years before doing my postdoctoral study in Dundee in Scotland for 6 years. Then in 2007, I started my lab in Tohoku University. Oops. So this is our institute in Tohoku University, Institute of Development, Aging, and Cancer, IDOC. And they are my lab members. Our institute is in Sendai in Miyagi Prefecture, where there are many touristic places such as Matsushima. But these coastal areas were severely damaged by tsunami following the devastating earthquake in 2011. And this is the year when we reported our first CHAMP1 paper. And at that time, it never occurred to me that this gene is related to intellectual disability. Since then, we published 6 papers as listed here, revealing various aspects of CHAMP1, which I will show you now. First of all, what is CHAMP1? CHAMP1 is the name of a gene and also a name of a protein. So gene is transcribed to messenger RNA you may have heard about in the COVID pandemic. This is a blueprint for a protein. And we have around 19,000 genes in every single cell in our body, which are on our genome. This is the total genetic information written in DNA sequence. And this genomic DNA is folded into chromosomes. The variation between individuals, which is the difference in DNA sequences, set around 0.1%. So this small variation makes difference between us, like the color of our hair. And some variations called mutations create pathological level of diversity. We have 23 pairs of chromosomes, one from father and one from mother. And CHAMP1 gene is located at the very end of chromosome 13. We usually use italic to show gene name. And from this CHAMP1 gene, CHAMP1 protein is formed. And CHAMP1 is short for chromosome alignment maintaining phosphoprotein, which I will explain later. And this is a schematic of CHAMP1. Protein is made of amino acid, and CHAMP1 is 812 amino acids long. And proteins can be divided into domains, which have different structures and functions. And CHAMP1 has zinc finger domain shown in blue in both ends, which form structures that bind to DNA, RNA, and proteins. And in this middle region, there are several repeat motifs shown in different colors. And these domains do not have a structure which is called intrinsically disordered regions. And this region is also phosphorylated at many sites, which affect protein structure and function. And proteins usually work by interacting with other proteins. 3 proteins are known to interact with CHAMP1. I do not go into deep with these 2, but Dr. De Andrea will talk about LEB7 in the next talk, and Dr. Baxter may mention HP1. So let me draw your attention on POX, which is a relatively new gene like CHAMP1, and there is a close relationship with CHAMP1, which I will explain later. And CHAMP1 interacts with POX at the tail region. And from 2015 to 2016, 3 papers reported CHAMP1 mutations in intellectual disability. And here, missense mutation and nonsense mutations and frame shift mutations are shown in different colors. And as you see, most of the mutations are nonsense or frame shift mutations, and most of them are de novo mutations. That means they are not inherited. However, in some cases, whole CHAMP1 gene is lost due to deletions, not by mutations, and some of them are inherited. You may already know about that, but let me explain types of mutation. When messenger RNA is used as a blueprint for protein, combination of 3 nucleotides, which is shown as A, G, C, and T, correspond to 1 amino acid, which is called genetic code or codon. And when mutation occurred, some nucleotides are lost or changed. And if corresponding amino acid is changed to another amino acid, this is called missense mutation. And some of the combination of nucleotides correspond to stop codon, which stop the elongation of amino acid. And if such stop codons are introduced due to mutation, this is called nonsense mutation. On the other hand, when some nucleotides are lost, the combination of these 3 nucleotides are totally changed thereafter. And as a result, sequence of amino acid is also changed thereafter, which is called frame shift mutation. And when it occurs, amino acid elongation stops sooner or later by the appearance of stop codons. Therefore, nonsense or frame shift mutation found in CHAMP1 give rise to truncated CHAMP1 that lack the tail region to varying degrees, as shown here. Importantly, POX, an interacting protein of CHAMP1 I mentioned, is also mutated in neurodevelopmental disorders. So this is a schematic of CHAMP1, which also has zinc finger domain, shown in yellow, and bind to CHAMP1 at its terminal region. And here, mutations are shown depending on whether they are seen in autism spectrum disorder, genetic intellectual disability, or both of them, or others, collectively called White-Sutton syndrome. This strongly suggests that CHAMP and POX may work together in neuronal development. And what happens when mutations occur in CHAMP1? There are a couple of scenarios. First scenario is these CHAMP1 mutants are not expressed because such abnormal proteins are often unstable because they cannot form proper 3-dimensional structure. The second possibility is that even if they are expressed, they do not function because they lack critical domains. So these cases are collectively called haploinsufficient. And because mutant CHAMP1 lacks the tail region, which is required for interaction with POX, they may not function even if they are expressed. The third possibility is that mutant CHAMP1 partially functions according to the sizes of the remaining part, which is called hypomorphic. This is less likely considering the severity of the symptom of the CHAMP1 individuals, which is not proportional to the sizes of deleted parts. The last scenario is this CHAMP1 mutant rather suppress the function of wild-type CHAMP1, which is called dominant negative. So we cannot exclude this possibility considering that symptoms of CHAMP1 deletion tend to be milder than CHAMP1 mutations. We examine whether these CHAMP1 mutants are expressed or not. We obtained lymphocytes from 3 CHAMP1 individuals and confirmed the mutations by ourselves. This is a result of a DNA sequence, and nucleotides are shown in different color, and mutations were detected at the expected positions. Then we examined whether these CHAMP1 mutants were expressed by gel electrophoresis, which separate protein according to their sizes. And this band corresponds to wild-type CHAMP1, and intensity of this band was reduced in lymphocytes from CHAMP1 individuals. And please take a close look at this band below the wild-type band, which is a mutant CHAMP1 with nonsense mutation at 649th glutamine residue. On the other hand, we cannot detect mutant CHAMP1 for other 2 samples. Therefore, we assume that these CHAMP1 mutants are expressed, at least in some cases, although we are not sure how they work. Let's move on to the next topic, the functions of CHAMP1. The first function is a role in chromosome segregation, which made us recognize CHAMP1 for the first time. Sorry for the busy slide, but I will guide you through it. Chromosome segregation is a process during cell division when the chromosomes, which are doubled beforehand, are separated into 2 daughter cells, as I show in this movie. And this process is carried out on the spindle, which is made of molecular fibers called microtubules. And chromosomes have to align to the middle of the spindle for proper chromosome segregation. And on these upper panels show the chromosome alignment of the normal cells, where chromosomes are shown in yellow and microtubules are shown in red. In contrast, when in lower panels, when we reduce the expression of CHAMP1 by small interfering RNA, as you see, chromosomes are misaligned because of the insufficient attachment to microtubules, which result in the chromosome segregation defect. And we found that the middle region containing a motif called the FP motif is responsible for this role of CHAMP1, and also found that CHAMP1 is highly phosphorylated in this region during cell division, and this is also required for the role in chromosome segregation. That is why we named this protein chromosome alignment maintaining phosphoprotein, CHAMP1 in short. We also found that CHAMP1 works in DNA repair. So the DNA in our body is continuously damaged by various insults. But fortunately, we can survive thanks to the many mechanisms that repair these damages. And among DNA damages, DNA double-strand breaks in which both strands of DNA, of the DNA double helix, are broken, and is the most severe DNA damage, and cells cannot survive if they are not repaired. So this graph shows the survival rate of cells exposed to ionizing radiation, which causes DNA double-strand breaks. And even normal cells die according to the increasing doses of irradiation. And when we reduce the CHAMP1 by 2 different small interfering RNA, so that this survival decreased further, showing that CHAMP1 is required to repair DNA double-strand breaks. And there are 2 pathways to repair DNA double-strand breaks. One is non-homologous enjoining, which is just like connecting the broken stick together with glue. And during this process, broken DNA pieces are often lost. Therefore, this repair is not accurate. However, in the second pathway, when DNA is doubled to prepare for cell division, cells can use this intact counterpart of DNA strand as a master copy to precisely copy the lost region for DNA repair. So this process is called homologous recombination, and we found that CHAMP1, together with POGs, promotes homologous recombination against inaccurate non-homologous enjoining. So the role of CHAMP1 and POGs in DNA double-strand break repair is also shown in other reports, and Dr. DeAndrea will describe it in detail in the next talk. Recently, we published the data on the analysis of CHAMP1 knockout mice. Gene knockout is a standard way to investigate the functions of genes of interest. Most of human genes have their counterparts in mice. Therefore, when we deplete genes, so the corresponding genes in mice by genetic manipulation, we can observe what happened in these mice when the gene is absent. In this paper, we acknowledge the CHAMP1 Research Foundation for continuing support of our study. From the following slide, plus means that both of CHAMP1 gene is present, which is called wild type, and plus minus means one of the CHAMP1 gene is lost, which is called heterozygous knockout, and the minus means both of CHAMP1 gene is lost, which is called homozygous knockout. And as shown here, CHAMP1 protein is lost in CHAMP1 homozygous knockout mice, and it is reduced in heterozygous knockout mice. And this table shows the number of pups obtained from the crosses of CHAMP1 heterozygous knockout mice. And according to the Mendel's law, the expected ratio of plus and plus minus and minus is 1/2/1. And the pups were observed at expected ratio until birth, but no CHAMP1 homozygous knockout mice survived to adulthood. And we found that CHAMP1 homozygous knockout mice die soon after birth. And at birth, the body weight of CHAMP1 homozygous knockout mice was slightly smaller compared to that of wild type and heterozygous knockout mice. However, other than that, they look pretty normal. So regarding the neuronal development, we observed the brain structure of CHAMP1 knockout mice. This is a section of brain of newborn mice. And here, we could not see clear difference between wild type and the CHAMP1 knockout mice. And when we take a closer look at brain structure at cerebral cortex, so there are 6 layers. And when we measure the thickness of each layer, again, we didn't see any difference between wild type and CHAMP1 knockout mice. However, we finally found the difference. This is important data, but a bit difficult to understand. So this is a section of cerebral cortex of developing the embryonic brain. And this green stuff is cells emitting light because they were introduced DNA to express green fluorescent protein 2 days before. And in developing brain, young cells called stem cells move from the bottom to the top while they become mature neurons. And so here, some cells reach the top. And after 2 days, this is 1 day before birth, many cells reach the top, forming layer structure, which I show in the previous slide. And when we reduce the CHAMP1 by introducing small interfering RNA together with DNA to express green fluorescent protein, you may find the number of cells reaching the top was reduced here. And even 1 day before birth, the number of cells reached the top, quite few in number, showing that migration and development of neuron is delayed when CHAMP1 expression is reduced. Therefore, although we cannot see big difference in brain structure at a glance, but there is a delay in neuronal development when we examine in such a detailed test. Okay? So in terms of symptom CHAMP1 individuals, we studied behavioral abnormalities in CHAMP1 heterozygous knockout mice, which represent haploinsufficient condition. 20 male mice, each of wild type and CHAMP1 heterozygous knockout, were subjected to behavioral test battery, as shown, as listed here, and the tests that show statistical significance were highlighted. For example, in T-MADE test, mice were placed in such a device and judged whether they turn to the right or left. And they normally turn right or left alternately. That means mice remember their previous decision. Okay? In 24-hour home cage social interaction test, 2 mice are placed together in one cage and observed how often they interact each other. And in post-third force swim test, mice are placed in water and observed how long they try to escape from water, so poor mice. Okay? Collectively, CHAMP1 homozygous knockout mice show mild memory defect, altered social interaction, and depressive behaviors resembling some aspects of the individual with CHAMP1 mutation. However, these differences are small, and we cannot tell the difference until we perform such a well-controlled experiment. Let's move on to the last part of my talk. After revealing various functions of CHAMP1, although I didn't explain the third one due to time restraint, our next question is how CHAMP1 plays these various functions. And the first possibility is that CHAMP1 plays these divergent functions independently. This is possible because many proteins are known to play different roles in different contexts. And it is also possible that other higher order functions, these cellular functions of CHAMP1 can affect neuronal development. The second possibility is that CHAMP1 plays these different functions through a common mechanism. And if there is such a mechanism, this would be a transcriptional regulation. So these two possibilities are not mutually exclusive. So it is possible that in some functions, CHAMP1 play a direct role, and for some functions, CHAMP1 works through transcriptional regulation. I'm sorry for this video slide, no one can see, but this graph shows the 94 genes related to neurodevelopmental disorders, and the CHAMP1 is here and the POG is here. And this vertical line represents the frequency of mutation of each gene. And among these 94 genes, nearly half of them are known to be related to transcriptional regulation, showing that the transcriptional regulation is closely related to pathogenesis of neurodevelopmental disorders. And so what is transcriptional regulation? So many proteins, including RNA polymerase II, play the role to regulate, to express genes at the right time and the right place by binding to the upstream region of the gene, which is called the promoter region. And also the distant part of DNA is also known to play a role in transcriptional regulation, which is called enhancer regions. So to address whether CHAMP1 plays a role in transcriptional regulation, we studied whether CHAMP1 localized to promoter and enhancer regions. This is one example of CHAMP1 localization on the genome. And here the localization of CHAMP1 is shown at the peak at the genomic region. And this is a map of one gene, and the transcription starts here. And the CHAMP1 localized around this transcriptional start site, which is a typical pattern of transcriptional regulator. So this pie chart shows the distribution of CHAMP1 around genome, and promoter and enhancer regions, so that here and here, accounting for nearly 40% of the total peaks, which is significantly higher than average, showing that CHAMP1 preferentially localized to promoter and enhancer region. And as you see, POX is also localized to this site together with CHAMP1, and we found that more than half of POX peaks co-localized with CHAMP1. So POX localization to promoter and enhancer region is consistent with recent reports. This is the last slide. So we found that these CHAMP1 peaks localized to 3,563 genes. Then we performed gene enrichment analysis to see to what kind of genes CHAMP1 preferentially localized. And we found that CHAMP1 peaks preferentially localized to genes related to intellectual disability. This adjusted p-value shows the significance of the result. The smaller, the more significant. And we also found that CHAMP1 peaks also preferentially localized to genes related to cell division, suggesting that so transcription regulation may also be related to the role of CHAMP1 in chromosome segregation. Therefore, our current hypothesis is that CHAMP1 POX may regulate expression of genes related to intellectual disability, and we are now pursuing this possibility further. Finally, I acknowledge all the collaborators for our research and current and past members of my lab, and these are funding bodies supporting our research. And last but not least, I want to acknowledge the CHAMP1 Research Foundation for supporting us and building such a strong scientific community. And I hope and I believe our efforts will culminate in the development of effective therapy for the treatment of CHAMP1 individuals in the near future. Thank you for your kind attention, and I'm happy to take any questions.
I'll start with the question, if you're okay. You mentioned before that wild-type proteins interacts with POGs, correct? Do we know any cases of where mutated CHAMP1 genes are still interacting with POGs, or they don't at all?
So if the tail region is maintained, I think they can interact with POGs. So in case of missense mutation, I think the tail region remained and can bind to POGs, but although we haven't checked in our hand.
Hi, that was really interesting. Thank you so much. Is there an inducible model of CHAMP1? In other words, if you had a haploinsufficient model, would there be a possibility of activating CHAMP1 after the mouse is born so you could actually see if gene therapy would be effective?
Thank you for the good question. The short answer is no. Short answer is no. Yeah, first of all, so that if you are talking about the CHAMP1 heterozygous knockout mice, first of all, it may be difficult to see the difference, even if we can make such kind of mice so that inducibly express CHAMP1. But first of all, I'm not sure whether we can see the difference, because CHAMP1 heterozygous knockout mouse itself looks pretty normal. I don't know so that it's answer to your question or not. On the other hand, we are now trying to make mice which can survive to adulthood, because at the moment, CHAMP1 homozygous knockout mice die soon after birth. So we cannot see what happened if CHAMP1 is lost in adults. So this we are now trying, and this can do this by inducible knockout, so that which we are now creating. Yes, thank you.
To Terry's point, it's really difficult working with mouse models when you're trying to translate drug development when they have very subtle phenotypes. I've worked with a number of programs in that space. But do you have a cellular phenotype in the mice? Because sometimes that's a lot easier to test the effect of drugs against if you have subtle behavioral phenotypes.
Could you make me clear? So what do you mean by salient mice?
So you showed delayed cellular neuronal migration. I think that was in cell lines.
Cell line. Okay, cell line.
Do you have a similar phenotype in the mice at the cellular level?
Cellular. We can try at least for other cellular functions. We can easily do this means called a rescue experiment. Rescue experiment means when we express the lacking gene externally, so that this is a kind of standard experiment to see whether the effect is specific to the gene. But so cellular level we can do, but at organismal level, it may be difficult at the moment.
So Kozo, thank you for wonderful talk. I have many questions, but I'm just going to ask maybe one or two, just because of time, and we can meet at lunch. So one question, or one point is, CHAMP1 is a DNA repair protein. You showed that it has a role in double-strand break repair. But very interesting, it also has a role in transcription. That's really interesting, your new data. And that's common for DNA repair proteins, that they have dual functions. They play a role in repair, and they bind to transcription complexes and regulate development. So do you know if the CHAMP1 protein it obviously binds to POG, does it bind to other proteins that are involved in transcription? Have you looked at other interacting proteins of CHAMP?
Okay, the question is whether CHAMP1 interact with other protein than 3 proteins I showed in the presentation. So we are searching for such protein for a very long time, and there are several candidates, and then there are. And then you can also see in the public database of the protein-protein interaction. And some looks promising, but so far none of them, there are some, but none of them have shown definitively for the relationship with CHAMP1. For this genomic localization, because of such kind of localization, many transcription factors co-localized with CHAMP1. But I'm not so there may be very transient and weak interaction between them, but maybe difficult to detect by conventional biochemistry.
Right. And one other question now, which is, we know that POG and CHAMP and HP1 alpha, they're involved in heterochromatin, certain regions of genome binding. When you did your chip analysis, did you also find POG and CHAMP at centromeres and telomeres and sites where there's heterochromatin?
Okay, that's a kind of professional question. So the HP1 is called heterochromatin protein 1. So on the genome, there's the heterochromatin and euchromatin, and so the heterochromatin is a rather inactive portion, and the euchromatin is the active portion. We first think because CHAMP1 and POX also bind to HP1, they may localize to heterochromatic region, but as I show now, so that it's rather opposite, so that it localizes to very active genomic region. But HP1 not only localized to such heterochromatic region, but also localized to euchromatic region, especially HP1 beta and gamma. So that this also Dr. Baxter mentioned. So that beta and gamma rather act so that not on heterochromatin, but also for act so that transcription regulation. No problem.
I have just two questions about the study, especially in the mouse. Amazing talk, of course, Kozo. Thank you very much. But do you see any difference in migration between glutamatergic and GABAergic neurons? Because sometimes, especially ASD, neurodevelopmental disorder, they have this kind of excitatory to inhibitory imbalance, and I'm wondering whether you see defects in at the cell type specific glutamatergic neurons or or inhibitory neurons.
We've never addressed this before, and yeah, I just want to collaborate with you on that point.
Okay, and another question related to this is whether you saw any kind of dendrites or spine differences between CHAMP1 knockout and the wild type.
We did observe so the length and the branching of spine, the dendrite, and also the number and the shape of spine. And so far, we didn't find any difference between wild type and that knockout mice. But we're not 100% sure that there is no difference.
Yeah, really elegant work, impressive. Now, for the phenotypic sort of features in terms of the depression on the swim test and the neurological symptoms that, be it that they may be mild, have you tried to rescue those sort of symptoms with gene therapy yet? And certainly the importance there is, what would be the timing to do so in such a model?
That is a very critical question, and which all of you want to know. So that to verify the effectiveness of some treatment, we need good model to see the difference. So as I said, so that this heterozygous knockout mice may not be a good model in such sense. I myself think but intellectual disability is not such a big problem for these inbred mice. So this may be the phenotype of the heterozygous knockout mice. But to see the difference more clearly, what we are trying now, and maybe others also are trying now, is to express, not delete the CHAMP1 gene, but express CHAMP1 mutant, so that which this idea depends on that, so that this mutant gene act negatively to the wild type gene, and so that for POGs, so that such kind of mice show more severe phenotype than normal heterozygous, just knockout mice. So this we are trying now. Is it?
Yeah, no, I think that's the sort of the key issue, I think, is that the phenotype that you see in the mouse, if you mutate CHAMP1, may not be exactly what you see in the human. But if you have a phenotype, and that's a consequence of the deletion of the gene or the loss of function of the gene, I think the important question is this: can you reverse that, even though the phenotype may not be exactly the same? I think that is sort of really the key to do that in an organism.
Yeah, I understand. So maybe for this sense, I don't have enough information. And I found that by talking with some of you, maybe I may overlook the phenotype of our mice, which can be corresponding to human symptoms, because I didn't know that CHAMP1 individuals have such a variety of symptoms. So we may take a closer look at this on some particular point to see whether our mouse model recapitulate the symptoms of the CHAMP1 individuals.
Okay, this is the last question, and we have to move on. Okay.
Also, you shared with missense mutation, you said amino acid substitution. Is it more of a substitution rather than a mutated gene?
So that your question is comparing missense mutation and the nonsense mutation, which showed the more severe phenotype, you mean?
Yeah. Yeah, yes, I would, because for a long time I was told it was all mutated for a missense mutation, but you said it was a substitution, so I'm trying to figure out what the difference.
Mm-hmm. The short answer, I don't know, but for the example of POGS, you see there are both missense and nonsense mutation, and so the POGS mutation causes both autism spectrum disorder and intellectual disability. And sorry. Sorry about that. You may see, this upper side is basically autism spectrum disorder. Then this lower side is intellectual disability. And the autism spectrum disorder tend to have missense mutation, while intellectual disability tend to have nonsense and frame shift mutation. We cannot definitely say autism spectrum disorder is milder than intellectual disability, but this suggests that, not 100% sure, but so the missense mutation may have a milder effect on our phenotype.
Thank you so much, Dr. Tanaka.